All right, welcome back. In this video, we're going to be looking at Unit 6, Lesson 2, Exploration 2. In our first exploration, we did a summary of this process that we call protein synthesis. And if you remember, protein synthesis has two stages. The first stage is transcription, and the second stage is translation, which we'll talk about in our next video. But for now, in Exploration 2, we are going to look at that first stage of the process called transcription. If you are transcribing something, then you are making a copy of it. You are writing down kind of the events that take place. The process of transcription is where we take that DNA molecule and we use it to create a copy in the form of RNA. Remember, our DNA molecule is very sturdy, it's very strong, it's built for long-term storage of information, and so we don't want to just take chunks out of it whenever we need to use it. We treat it like a cookbook, where we open it up, we go to a specific location, and we transcribe or we copy the information into a temporary storage molecule of RNA. This would be like, creating a Post-it note or a sticky note of information from a book that we want to be able to use. All right. Now, in eukaryotes, which are cells that have organelles and nucleus and those sorts of things, this process takes place inside of the nucleus of that cell because that's where the DNA is located. All right, so let's look at how this actually happens. When a cell receives a signal that we need to make a protein, the DNA of that cell opens to a specific location where that gene for that protein is found. And as that DNA opens up, a molecule called RNA polymerase initiates this process of building RNA. Now, if that word polymerase sounds familiar, it should. We introduced you to the idea of DNA polymerase when we talked about replication. If you remember, DNA polymerase is responsible for building new DNA. As you can probably imagine, RNA polymerase is responsible for building new RNA. It adds new nucleotides to an RNA molecule in a complementary way to the existing DNA strand that it's looking at. So, back to our picture. We open up the DNA at the location where the gene for a protein is found. The RNA polymerase begins adding those RNA nucleotides in the order in which case they need to be put together to build our protein. And once we get to the end of that gene, the RNA molecule pops off and now can be sent to the cytoplasm to be built. Now, a couple things. Before this RNA molecule is ready to go, it has to be processed, meaning we kind of package it up so that it's a little bit safer. We remove any unnecessary sections, and we make it about as simple as it possibly can. We don't need all of the extra stuff that's sometimes found in DNA. We just need the information specifically for the protein that's about to be built. All right, so this set of three pictures is pretty useful to look at to understand how this happens. Now, this animation is another pretty cool animation. When you look at this, I want you to be thinking about a couple things: the DNA opening up, the mRNA being formed, and then ultimately the mRNA leaving to go be built. What you're seeing here is this section of DNA being transcribed into an RNA molecule. This yellow string would be that RNA. And all of these little yellow flecks around it are the different nucleotides that are being pulled in and added to that RNA structure as it's being built. Now, some of these RNAs that we're building, these what we call messenger RNAs, they can be really long. Some of them are really short. It just kind of depends on the type of protein that we need. But this blue structure right here would be that RNA polymerase that's able to open it up, read it, and then add those new spots to it. as we get closer to the end. Once this mRNA strand is finished, it ultimately is going to be released so that, again, it can be processed and made ready for the process of building a protein. There it goes. Now again, in transcription, we're going to make lots of copies of these genes because we might need lots of different proteins. The cool thing about this process is that once it starts, almost immediately another RNA polymerase can attach on and start building a new messenger RNA for a new protein. All right. Now, once that mRNA is built, it's going to leave the nucleus and attach to a ribosome where it can then be built into a protein. Now, this next diagram is going to show you a couple of the different types of RNA that you're going to need to be aware of. If you can keep track of these 3 types of RNA, then this whole process becomes much simpler. Okay, first of all, this is the mRNA. This is your post-it note. This is your temporary storage of information. The mRNA then goes and attaches to an rRNA, which is a ribosome. And that ribosome, as we talked about last semester, is used to build proteins. Now, tomorrow, we'll talk about how the tRNA, the transfer RNA, is responsible for helping to then build our protein from the instructions that we just copied from the DNA. All right. Again, next part is translation. We'll talk about that in our next video.